Imagine this, you haven't even eaten anything for almost 24 hours. There's no glucose coming from food, and your liver's glycogen stores are almost empty. But your brain is still working. Your red blood cells are still alive. Your kidneys are still functioning. So, where the glucose come from? Is the body creating glucose out of thin air? Actually, yes. The body starts manufacturing brand new glucose from the substances that aren't carbohydrates. That amazing life saving process is called as the gluconeogenesis. And once you understand this lecture, you will never confuse gluconeogenesis with glycolysis again. Before we begin, let me tell you why this chapter is so important. Almost every university asks questions from the gluconeogenesis, not because they want you to memorize enzymes, but because this pathway explains what happens during starvation, diabetes mellitus, prolonged fasting, and severe infection, trauma, and critical illness. Whenever a patient survives without eating for days, the hero working silently inside the liver is the gluconeogenesis. So today, don't memorize, understand the logic, and once the logic is clear, the entire chapter becomes easy. Let's start with the name itself. Gluconeogenesis. Let's break it down. Gluco means glucose, and neo means new, and genesis means formation. So literally formation of the new glucose. But here's something students often misunderstand. Is the gluconeogenesis simply the reverse of glycolysis? No, it is resembles the reverse pathway, but it's not exactly the reverse pathway. Will it see why it shortly? The definition is the gluconeogenesis is the synthesis of the glucose from the non-carbohydrate precursors. Notice the words non-carbohydrate precursors. That means the body is producing glucose without using dietary carbohydrates. This is one of the favorite university definitions. Don't forget the word non-carbohydrate compounds. But wait, if carbohydrates are absent, where does the body get the raw material? It's the excellent question. Let's answer that. These are the substances the liver converts into glucose. These are the five major precursors. First one is lactate. Where does lactate come from? Think of the person running the 100-meter sprint. During the intense exercise, muscle don't get even oxygen, so they switch to anaerobic glycolysis. Anaerobic glycolysis produces the lactate. Then we will see the pyruvate precursor. What happens if pyruvate isn't required for energy production? The liver simply diverts it and towards the glucose formation. Think of the pyruvate as a traffic junction. It goes towards TCA cycle, lactate, alanine, and gluconeogenesis. The body chooses whichever pathway is needed. Then the glucogenic amino acids. Now imagine you have been starving for 2 days, and where will the glucose come from now? The body begins breaking down the proteins. Proteins become the amino acids, and many amino acids are converted into glucose. These are called the glucogenic amino acids. Here's the one memory trick that the glucogenic gives the glucose. Remember the one important exception, leucine and lysine are not glucogenic. They are purely ketogenic. This is the another favorite exam question. Glucogenic amino acids are the most of the amino acid, but there is exception leucine and lysine. Remember as L2. And glycerol is the one of the precursor. Every triglyceride contains one glycerol, three fatty acids. When fat breaks down, fatty acids and glycerol separate. Students often think fatty acid become glucose wrong, and fatty acid generally cannot produce glucose, but glycerol can. And finally is propionate. This is the small but important exam point that the certain fatty acids which an odd number of the carbon atoms produce, that is the propanyl CoA, this is converted into propionate and eventually the glucose. Not very important physiologically in humans, but very important for university exams, never ignore it. Let's see about the location of the gluconeogenesis. Where is glucose manufactured? Mainly in the liver. And liver produces approximately 1000 grams of glucose per day under the normal conditions. Liver is the major organ, but the liver is the only organ? No. The kidneys, especially during the prolonged fasting, also performs gluconeogenesis. This is why patient can survive prolonged starvation. Inside the cell, most reactions occur in the cytosol. However, some important initial reactions begin inside the mitochondria. We will understand further. If gluconeogenesis is energy consuming, why doesn't the body perform it all the time? Because it's expensive and the body first uses food glucose and then glycogen, and only then gluconeogenesis starts becoming significant. The body always saves energy whenever the possible. Let's see about the importance of the gluconeogenesis. Now comes the heart of the chapter. Why do we even need this pathway? Let's understand logically. Supplies of the glucose to glucose dependent tissues. Some tissues have almost no choice, they must use glucose. That example, brain and central nervous system, red blood cells, and kidney, medulla, and testes. The brain alone consumes 120 grams of the glucose per day. An entire body requires around 160 grams daily. Imagine fasting for one day, dietary glucose is absent. Without gluconeogenesis, blood glucose would fall and the brain cells would stop functioning and the life would be threatened. That is why the gluconeogenesis is literally survival mechanism. Next, the during intense exercise, muscle produced lactate and the liver converts lactate back to the glucose and the glucose again returns into the muscles and the body is constantly recycling the fuel. We will learn in Cori's cycle later. After prolonged fasting, liver glycogen becomes exhausted. Now the body has only one major way to maintain blood glucose, that is the gluconeogenesis. Without it, human cannot survive the prolonged starvation. This is an extremely important conceptual question. Here is the substances like lactate, glycerol, and propionate accumulate continuously instead of allowing them to build up. The liver converts them into the glucose. Nothing is wasted, and nature is an excellent recycler. In critically ill patients such as severe injury, burns, infection, trauma, here gluconeogenesis increases markedly. Why? Because stress hormones increases and the body desperately needs the glucose. Hence, body needs glucose. Unfortunately, the excessive gluconeogenesis can worsen the hyperglycemia. Then, excessive gluconeogenesis contributed to the elevated blood glucose. Persistent hyperglycemia can impair and the immune function, wound healing, blood coagulation, and increases the oxidative stress by the generating free radicals. This is one of the reasons why uncontrolled diabetes becomes dangerous. So far, we have understood the what gluconeogenesis is, why it is essential, where it occurs, and the major precursors. But now comes to the most important and most frequently asked university exam concept, that if gluconeogenesis is the reverse of glycolysis, why can't it the body simply run glycolysis backwards? The answer lies in three irreversible steps. Once you understand three reactions, you have mastered almost half of the gluconeogenesis. Let's think logically. Glycolysis has 10 reactions. Out of these, 7 reactions are reversible and 3 reactions are the irreversible reactions. These irreversible reactions are the catalyzed by hexokinase, phosphofructokinase first, and the pyruvate kinase. Now here's the problem. The irreversible means you can't simply run them backward because that would require enormous amount of energy. So what does nature do? Instead of reversing those reactions, it creates the alternative pathway called as the bypass reactions. Think of it like a road. Imagine a bridge is permanently closed. Can you drive it through it? No. You need the detour. Exactly the same thing happens in gluconeogenesis. 3 roadblocks while glycolysis backwards. Hence, in the gluconeogenesis, uses 3 bypass reactions. Here are these 3 bypass reactions are there. These are the alternative enzymes specific to the gluconeogenesis. First one is conversion of pyruvate to phosphoenol pyruvate, And next one is conversion of fructose to 1,6 bisphosphate, and third one is the conversion of the glucose 6 phosphate to the glucose. These are the reversible steps of the glycolysis. Let's discuss one by one in the deep. Bypass reaction first is the pyruvate converted into phosphoenol pyruvate. This is the longest and most interesting bypass. Pyruvate becomes oxaloacetate, and pyruvate carboxylase is the enzyme is used. Pyruvate cannot directly become phosphoenol pyruvate, hence it must become oxaloacetate, the enzyme responsible for the pyruvate carboxylase. Here is the pyruvate carboxylase. Ask yourself that where is the enzyme located? It is in the mitochondria, and that's why the first reaction begins inside the mitochondria instead of the cytosol. These enzymes require ATP, carbon dioxide, and the biotin. Biotin acts as a carrier of the carbon dioxide, and without the biotin the reaction cannot occur. Here is the equation that pyruvate requires the ATP. Another favorite question is that pyruvate carboxylase is activated by the acetyl CoA. Why? Because imagine lots of fatty acid breakdown. Fatty acid produce acetyl CoA, and the high acetyl CoA tells the liver we already have enough fuel. Instead of burning pyruvate, convert it into the glucose. Very logical. But wait, oxaloacetate is now formed inside the mitochondria. Can it simply leave mitochondria? No. This is where the students become confused. Here is the required the malate shuttle. Oxaloacetate cannot cross the mitochondrial membrane. So what does the cell do? It temporarily converts the oxaloacetate into the malate. Malate can easily cross the cytoplasm, and once it reaches to cytoplasm, it becomes the oxaloacetate again. Think of these like a changing trains. The passenger cannot cross directly, so he changes vehicles. Oxaloacetate changes into malate, crosses the membrane, and then changes back into the oxaloacetate. Here is the step 1 that oxaloacetate converted into phosphoenol pyruvate. Now another enzyme comes, that is the PEP carboxylase. Here is the PEP carboxylase. Here is the, what does it need? GTP, not the ATP. Remember this difference, during this reaction carbon dioxide is removed and GTP provides energy. Students often ask, sir, gluconeogenesis is expensive? Very expensive. Just this first bypass requires GTP, and that's why the body performs gluconeogenesis only when absolutely necessary. Next bypass reaction, second one is the fructose 1,6 bisphosphate is converted into fructose 6 phosphate. Now the pathway comes reverse, and PEP undergoes the several reversible glycolytic reactions until becomes the fructose 1,6 bisphosphate. Now comes the another roadblock, that is the fructose 1,6 phosphofructokinase cannot run backwards, so another enzyme is required, that is the fructose 1,6 bisphosphatase. Notice the name carefully, not a phosphofructokinase, fructose 1,6 bisphosphatase. It removes the phosphate, and here is the requirement is magnesium. This enzyme is one of the major regulatory enzymes of the gluconeogenesis, very important. This enzyme is absent in skeletal muscles and cardiac muscles. Because muscles don't release glucose into the blood, their job is to use glucose, not manufacturing the blood glucose. Students mistake. Many students confuse that PFK1 phosphofructokinase first with the fructose 1,6 bisphosphatase. Remember, one belongs to glycolysis and other one belongs to gluconeogenesis. These are opposite pathways and opposite enzymes are there. Third bypass reaction is the glucose 6 phosphate converted into the glucose. Now only the final step remains, that the glucose 6 phosphate must become the free glucose. These enzymes responsible for the reaction, that is the glucose 6 phosphatase enzyme. It removes the phosphate group. Finally, the free glucose is produced. Where is the enzyme is present? Mainly in the liver and kidney, absent in skeletal muscles, brain and adipose tissues. Now think logically. Why can't skeletal muscles release glucose into blood? Because it lacks the glucose 6 phosphatase enzyme. Muscles can store the glucose as a glycogen. Muscle can use glucose, but the muscle cannot export free glucose. This is one of the favorite university viva question. And the overall energy requirement for these reactions are the 4 ATP, 2 GTP, 2 NADH. And this tells us one thing: gluconeogenesis is not a free, it is the energy consuming investment made by the liver to keep the vital organs alive during the fasting. Energy requirement is 4 ATP, 2 GTP, and 2 NADH. Let's discuss about the next part. First, we will see one diagram for the gluconeogenesis. Here is the reverse pathway, here is the red is the glycolysis and blue is the gluconeogenesis. It starts from the pyruvate, then converted into phosphoenol pyruvate. It is the mediate that oxaloacetate. These are the two enzymes. Here is one irreversible pathway. Here is one reaction, another reaction, and here is the third reaction. Let's see the Cori cycle and Cahill cycle. Glycogenesis form from the amino acids, from glycerol and from propionate also. But here is one important from the lactate. Think of the liver as a recycling factory. Every tissue sends its waste products to the liver instead of throwing them away. The liver converts them into fresh glucose and nothing is wasted. This is one of the smartest survival mechanisms of our body. Let's understand each precursor one by one. Let's see about the lactate. Here, imagine you are running a 400-meter race and your muscles are contracting continuously. Very soon, oxygen supply becomes insufficient. Without enough oxygen, muscles switch the anaerobic glycolysis and glucose becomes pyruvate, but because of the oxygen lacking, pyruvate cannot enter TCA cycle. Instead, it is converted into the lactate by the enzyme lactate dehydrogenase. Then lactate accumulates inside the muscles. If too much lactate accumulates, the muscles become acidic and this reduces the muscle performance. So what does the body do? The lactate leaves the muscles, travels through the bloodstream and reaches to the liver. Now inside the liver, lactate is converted back into the pyruvate by the LDH, and through gluconeogenesis, pyruvate becomes the glucose. Finally, it returns to the muscles and used again for the energy. This entire cycling process is called as the Cori cycle. Why the Cori cycle is important? It has the three major functions: removal of the excess lactate from the muscles, prevent lactic acidosis, and supplies the fresh glucose back to the muscles. Hence, think about glucose recycling system between the muscle and the liver. Here is the one diagram for the cycle, Cori cycle. Here is the muscle, lactate is formed because of the anaerobic glycolysis, then the going to liver and finally forms the pyruvate. It enters into the gluconeogenesis and glucose is formed. It's over. Let's see about the Cahill cycle. This is the theory about the Cori cycle and this is the Cahill cycle. During prolonged fasting, muscles start breaking proteins and these proteins produce the amino acids. Inside the skeletal muscle, pyruvate combines with amino groups and to form alanine. Alanine enters the bloodstream, travels into the liver. Inside the liver, alanine loses its amino group and becomes the pyruvate. And the pyruvate enters the gluconeogenesis. Finally, glucose is produced and the glucose returns to the muscles. So cycle transports carbon skeletons for the glucose production and amino groups safely from the muscle to liver. Unlike the Cori cycle, which transport lactate, and the Cahill cycle transport the alanine. Here is the one easy comparison between Cahill cycle and Cori cycle. Cori cycle where lactate is transported, in Cahill cycle alanine is transported. Cori cycle anaerobic exercise due to and Cahill cycle due to prolonged starvation. Cori cycle removes lactate and Cahill cycle removes the amino nitrogen, and it is both occurs in muscle and livers. Let's see about the regulation of the gluconeogenesis. Here is the also alanine is going to liver, pyruvate form and enter into gluconeogenesis and glucose is formed. This is the Cahill cycle. So simple. So far we have answered the three major questions, that what is the gluconeogenesis, how does it occur, where do its substrates come from. But here is the final question: who decides when gluconeogenesis starts? Because imagine the something impossible. If glycolysis and gluconeogenesis ran together at a full speed, the cells would simply waste ATP without producing anything useful. So the body needs a very intelligent control system. That's what we are going to learning today. Why regulation is necessary? Let's think logically. Suppose you just finished a heavy meal and blood glucose is already high. Should the liver make the even more glucose? Absolutely not. Now imagine you have been fasting 24 hours. The should gluconeogenesis start? Definitely yes. So the body continuously checks the blood glucose levels and adjusts gluconeogenesis accordingly. This regulation mainly depends on hormones and availability of the substrate and the energy status of the cell. Three are the major regulators: hormones, availability of substrate, and acetyl CoA. Three important hormones are the glucagon and the glucocorticoids and the insulin. First, talk about the glucagon. When the glucagon released? After eating? No. Glucagon is released during fasting, starvation, and a low blood glucose. It is secreted by the alpha cells of the pancreas. Hence, glucagon acts through two important mechanisms. That is, first, that glucagon increases the cyclic AMP, and this converts active pyruvate kinase into inactive form. Now think carefully. The normally pyruvate kinase converts PEP, phosphoenol pyruvate, to the pyruvate. But when the pyruvate kinase becomes inactive, PEP is no longer converted into pyruvate. So where does PEP go? Towards the gluconeogenesis. Instead of being used in glycolysis, PEP diverted for the glucose synthesis. Glucagon blocks glycolysis and pushes the gluconeogenesis. Remember this. And second mechanism, that now comes another favorite exam concept, that the glucagon decreases the concentration of fructose 2,6 bisphosphate. Fructose and students often confuse with this fructose 1,6 bisphosphate. These are the completely different molecules. Don't mix them. Role of the fructose 2,6 bisphosphate is in the activate the phosphofructokinase 1 in glycolysis and inhibit is gluconeogenesis. Next one is the glucocorticoids. Whenever the body experiences trauma, surgery, severe cases like burns, starvation, glucocorticoids increases. But why? Because stress requires the energy and this releases amino acids. Those amino acids become substrates for the gluconeogenesis. In simple words, more amino acids, that the more glucose production. This is why the patients receiving long-term steroid therapy often develop hyperglycemia or the steroid-induced diabetes. Now think about the insulin. Insulin is, after eating, the blood glucose rises and insulin is released. Should gluconeogenesis continue? No. Insulin suppresses the gluconeogenesis. How? It reduces the activity of key gluconeogenic enzymes. So remember one golden rule: insulin is a storage hormone and glucagon is a fasting hormone. Whenever you forget, just think about the fasting versus feeding. Everything becomes easy. Second one is the availability for the substrate. Even if all hormones say make a glucose, can the liver do it without raw materials? No. Substrate must available. That major substrate include lactate and glycerol, glucogenic amino acids and propionate. During uncontrolled diabetes, insulin deficiency causes muscle protein breakdown. Large amounts of amino acid reach to the liver, more substrate and more gluconeogenesis, more hyperglycemia. This creates the vicious cycle. Then the role of the acetyl-CoA. Finally, acetyl-CoA during the starvation fat breakdown increases. Large amounts of acetyl-CoA accumulates in the liver. Acetyl-CoA allosterically activates pyruvate carboxylase, and remember, this is the first enzyme that first bypasses the reaction. The activation of pyruvate carboxylase increases the gluconeogenesis. Let's revise. Major precursors are the lactate, pyruvate, gluconeogenic enzymes, gluconeogenic amino acids, glycerol, propionate. Major site is liver, kidney during the prolonged fasting. Three bypass reactions are the pyruvate oxaloacetate to PEP. Second one is fructose 1,6 bisphosphate converted into fructose 6 phosphate. And the third one is glucose 6 phosphate converted into liver. Energy requirement is 4 ATP, 2 GTP and 2 NADH, and cycles are the Cori cycle for the lactate, Cahill cycle for the alanine. Regulation is that increases favors by the glucagon, glucocorticoids and acetyl-CoA and inhibited by the insulin. Here is we covered the gluconeogenesis. So that's complete concept of the gluconeogenesis from definition, importance, from precursors, bypass reactions, Cori cycle, Cahill cycle to regulation and clinical correlations. Don't try to memorize isolated enzyme names. Instead, remember the logic when blood glucose falls and liver becomes the glucose factory. If this lecture is helped you truly understand the concept rather than just memorize it, subscribe because we covering another topics also. And thank you.